lots more people coming in. We had tons of sign up for this panel. I'm really looking forward to getting started. So I'm going to do a quick intro to get us kicked off here. So I'm Oliver Gaucher. I work with climate farmers on the academy side, and this is a production of the nonprofit wing of this organization, where we are trying to bring a community of farmers all over Europe together in order to share knowledge and have access to what's going on in the field and the cutting edge information that's available to help them succeed in their own endeavors. And that's what brings us here today with Harriet and James. We're going to be exploring the cutting edge of soil science and agronomy from some of the people who are at the forefront of this type of research. And, well, I'll let you each introduce yourselves real quickly. So, Harriet, would you like to give us your intro to get us started?
I'm here. Okay, I am a biologist. I studied in Tübingen in ages ago, and I work with soils. I work with seeds. I work with people. I like education, and I do product development. And I think that should do.
Perfect. We'll get into the stuff that you're excited about and researching at the moment in just a minute. James, can you give us a short introduction on yourself and your work?
Yes, but I'll add one more thing about Harriet. She has a command of plant physiology like nobody I know. So she knows she's being somewhat modest. Sorry, Harriet. Sorry about that. I'm James White. I'm a professor at Rutgers University in New Jersey. I work on endophytes, and I teach courses here on fungi and plant science and other courses over the years. And I think that's it.
Well, perfect. Let's get started with the first question of our panel today, which is, what information or new discoveries do you find to be most exciting in the areas of soil science and agronomy at the moment, and why? Harriet, would you like to start us off?
Oh, it's me.
I'm going to pick on you all night, so.
Oh my God. Latest discovery in soil science. For me, it's maybe it's this. Maybe it's to shift plant or agriculture around from that view that we're source limited to sink limitation. So we have this true deficiency state when the soils are wrecked and when the plants are in need of minerals and everything, and nutrition is bad and whatever. And we kind of can insert this through several tricks and shortcut the system and And then comes the moment where the system starts to stagnate and you have this feeling, what's going on? I'm doing my practices, I'm fertilizing, and I'm, and the system doesn't leap, it doesn't fly. And the understanding that all these things are working in feedback loops, and if the product is not used, that we go into feedback inhibition of production, and that we sit there and have the plants sitting in the furrow and maybe not doing what they should do, not feeding, not producing, because we broke the cycles. So basically, to come to this idea, to understand that a system has to deserve its status itself, and then understand that when the demand is in the whole system and the cycles are closed, that's when it starts to really hum. So, for example, to be concrete, we have photosynthesis. Photosynthesis produces carbohydrates. If these carbohydrates cannot be extracted from the leaves and they accumulate there, they are a problem. Production needs to shut off. If they go and are exported into the root and they are not consumed down there because we have too little oxygen and the microbiome around the root is not consuming that, then these things go back up in the shoot and they switch photosynthesis off. Same thing. And So we have this idea we can maximize photosynthesis and again impose our will on this plant, but in fact we can't. So understanding what we have to do all along this loop so that production and consumption in growth through the microbiome work steadily will just really relieve this system and help the production go steady and production be boosted to another level. And this is this idea to go away from from source limitation to sink limitation, and also go away from this idea of precision control of everything to this idea to understand and be part of a system and get trust in the system to work. And I'm sorry, this is not mainstream soil science, because this goes into the direction of control and manipulation. Because every paper you read is like, oh, we found the super tweak now, and we're going to make this wow effect and be better than creation intended it. But there is this other trend, which is usually away from these publications, where you see people gain trust and gain a handling of the systems. And this excites me. If that really works, very good.
Fantastic. That's the type of answer that I was counting on from you. James, I'm going to pose the same question to you before we dig deeper into this system understanding of plant and soil health. Can you tell us about what you're most excited about at the moment on the forefront of your own research, or what you're excited to be digging into this upcoming year?
I will. And it's something that we are just beginning to, in science, get an inkling of, and that is that bacteria, for example, soil microbes have especially bacteria have a huge effect on the plant, and the possibility is that effect is directly on the genes of the plant, causing additional genetic variability in the plant, and some genetic changes that affect plant expression, the expression of the plant's genome, right? So epigenetics. So that's for the plant, but now talking about soil science, I mean, that's involved there because those microbes are from the soil that the plant is getting. But the other effect is on another group of organisms, and those are the fungal pathogens. And they also will associate intimately with bacteria, including inside their hyphae, and vectoring in their spores, for example, when they make spores. And those associations from experiments, we know those associations affect the behavior of the fungus, a fungal pathogen, and at least in many cases, the virulence is reduced when they have bacteria there. There's a few cases where it goes up, but generally it goes down. And so the really what is really new here is beginning to try to sort out how these interactions are happening that favor the plant development, both inside the plant, but also in the soil environment, and potential pathogens in the soil. So ultimately understanding the genetics of how that happens. So that's what I find. To me, that's the most interesting area right now.
That is fascinating. And I'm actually curious, Harriet, how that fits in with what you were talking about, because James basically outlined some complex relationships and the relationships between different organisms within the soil microbiome. And this seems to me to fit into this larger systemic idea of relationships and cycles, which you were mentioning.
Well, I've spent the last years on some mental hobbies, and one of these is sorting the frame set of interaction of the endophytes with the plant. And what fascinates me really is that you can look in any layer, and for me, it's just the more I know, the more ridiculous I find it to hijack this system with a single compound or something. Because it's like all these safety layers, like for example, plant, when it has to recognize it is a friend or is it an enemy that's just sitting in my roots, or is it, or how they control, for example, a mycorrhiza, that it just doesn't grow and drain nutrients, but really work for what it's getting there. And then you see on every organizational level, be it the genes, be it the minerals, be it the chemical communication, be it the structure, it's just interlaced to a degree where you sit there and it's like, wow, it's absolutely fantastic. And then we come along and we think, okay, we just take a plant hormone, like we use auxin and we just pour it on the plant and it becomes bigger. And then it's like, well, yes, more biomass, but has it the minerals or not? And then you find out, okay, you have root microbiome that gets certain amino acids from the plant, and then it transforms them into auxin, and this is taken up from the plant, and then you have these growth effects. And then you think, well, why do they do it? And then you see that the plant, to produce this amino acid, takes zinc. And if it has zinc sufficiency, it will do this, and then you have this whole cycle. And where does the zinc come from? It comes from the microbiome again. And so you see that all this is interlaced on so many levels of interaction, and it must be so. And the big question that I have is, you have all these players there, if you have them. I hope you have them. You have all this diversity of bacteria. You have all these metabolic cycles. You have all these fungi there and everything. And it can immediately go into any direction. All these big circles, to volatilization, to leaching, to feeding the plant, fixation. And the question for me is, why and how does this result in growth? And why and how does this result in destruction? Like James says, you have these bacteria and they go in the hyphae and wow, they're not virulent anymore. But why do they do so? And then we have these super smart people that come along now and say, okay, we just forcefully inject some super bacteria in these fungi, and then we have this super fungus, and then we put this out there and our system is going to make woof. I doubt it. Or manipulate, for example, there is a very new paper about the recognition mechanism. What is a symbiosis and what pathogen? It's like a symbiosis results in very even, rather slow calcium oscillations in the inside of the plant, coming from the nuclear region. And if you have a pathogen, this goes very fast and irregular, or if you have stress, it's just all messed up. And okay, you can go on now and you say, we manipulate this endogenous rhythm then. And then we have a plant that's going to take up the symbionts more readily. Super. And our wheat that is so hard to go along with mycorrhiza, now it will be handshaking, much easier, and it will be working with the mycorrhiza. And I look at this and I think, folks, maybe, but maybe not, because it is just interlaced in so many layers, and you have all these metabolic pathways linked to this, and all these signaling mechanisms and everything. You're going to produce another problem on the other end. And I think, why don't we instead invest the energy for this quest on how to get these organisms really settled in the soil and how to let them interact there in a friendly way? This would what I would like to see in terms of research, more, much more.
How to create healthy soils, right? How to create good regenerative soils.
Plant-friendly soils.
Yeah.
Yeah,
And what,
how does that
what
fit in with what
means-
you were talking about? How does that fit in with the relationship between the pathogenic microbes and the endophytes and the relationships that you outlined earlier? Does that fit in with the model that you're investigating right now?
meaning the model that we're discussing, looking at relationships?
Yeah,
Yeah.
exactly. Instead of manipulating
Yes.
individual parts, looking at it as
Yeah.
this larger ecosystem.
Yeah, I think that's the key, really. I think the ecology of it is, and that's the nice thing about regenerative agriculture, is that growers really have to be ecologists. They have to think like an ecologist. And this is basically what we're talking about. And it's not just, we're talking about, we have to think about plants differently, plants as organisms, plants and fungi that exhibit behaviors, right? You look at their behaviors and you see, with this microbe in the system, then the behavior changes or it doesn't, right? How does that behavior change? So you can look at these interactions that are happening between the microbe and the plant. And some of those endophytes, some of the bacteria that plants take into them, actually go out and also affiliate with fungi in the soil. So even the same organism can create a kind of a bridge between potentially pathogenic fungi in the soil and plants, and alter that association in between between the fungus and the plant. So, and that's fairly common. It's not like an uncommon observation. It's a frequent observation that bacteria will change behavior of fungi. A lot of people, when they look for these kind of things, they'll grow them in culture and they'll just make a little colony of the bacterial colony, then they put the fungus near it, and they look for things that inhibit, right, that make the fungus go away. But that's really not how it works in nature. In nature, mycelium is growing and bacteria will grow right on the surface of them and internally colonize as well. So that's what happens in nature. It doesn't necessarily kill the pathogen. It just alters its behavior, reduces its virulence, or completely eliminates its virulence. And some fungi with bacteria with them, some pathogens become endophytes because they're no longer virulent. And they actually benefit the plant development. So that whole approach, looking at relationships, yes, I think that's important. And behaviors, that's the other thing. Farmers, growers can, we're really looking at their crops. They're looking at how their crops are behaving, how they're growing and, what's happening out there. So, yeah, I think that they're focusing on relationships, ecological relationships is a key thing.
I really want to go deeper into what you started talking about here, which is how this information, which probably is going over the head of quite a few people, myself included at times, can be translated into practical understanding and action out in the field, right? So with all of these things at the cutting edge of research and this greater understanding of an ecological point of view of what is happening at a microbiological level, how does this translate into action in the field, and what can people do with this understanding when managing their farms and their crops?
Harriet, you want to do that first? First? You got any ideas? Okay.
Basically, what I see is that we currently have two profitable, actually, possibilities to look at these things. Number one is to create super specific, highly controlled, precise environments and play with these ideas. So you have the highly selected plant that just works with this mycorrhizae, with this bacterial species sitting on this mycorrhizae and in this soil, and with this irrigation and 3 microliters of water on the right leaf. Like, so, this idea. And then, I don't know, high voltage over the head to simulate the thunderstorm in approach like in China. All these things just really. So that's the one thing. And the other thing is to say, I look at it in terms of a local evolution. And I allow the system to self-optimize and to learn. And I entrust myself as part of the system to the idea that it is a self-optimizing system. Because if you look at what Toby Kears, for example, has found, that mycorrhizae, they are trade agents and they go with their materials where they get paid. and they just really shift them from sources where they have high phosphorus to places where there is little phosphorus, and they really bargain for it. They know what a young root is, they know what an old root is, and they go for the host that really pays well. What does that mean? That means that if we have several generations in a certain condition, that the best paying host will have a growth benefit, and this host controls different mycorrhizae around its root, so it selects and feeds the best one most. So this is like a Munchausen system. It's an upward spiral. And that means then when we close these loops again, then when we let it run for several generations, and then we come back to James, we had just recently a discussion after my mycorrhizal talk about the change in amino acid composition, because I wanted to know from him if he was into that. And he said, yes, of course. It's not just the root that enriches and lies in the methionine, it's the shoot as well. And when you look into physiology, you see that methionine, again, it just changes, really literally chemically changes the DNA. It changes the gene activity. So we have this interplay going on. And if we let this run for several generations, so the key is that we really dare to let it happen. And when we
She seems to be freezing right at the most crucial moments. James, can you jump in there for a second?
Yeah, no, I can put this, okay, this concept of the self-optimizing system, soil microbes and endophytes play a big role there. And basically these plants take the microbes out of the soil that they need to survive in that environment. So they essentially adapt, they optimize to that environment, but you have to grow them for a while to do that. So that's a little practical thing there, so one of the few practical things that I could think of.
So the concept here is to take more of a hands-off approach if what you're used to is trying to micromanage the balance of nutrients or the presence of certain life forms in the biology. Is that what we're getting at?
Yeah, the point is you build your soil, you build your community so that the plants can actually take care of themselves, so that I mean, yeah, we have one concept, chemical industrial agriculture, where we control everything. We're controlling that environment. We're doing it a certain way. It's very technician type focused, right? Just everything has to be technically done. But then there's the other way, the regenerative way. Okay. And that is this self-optimizing system that uses microbes and nutrients naturally available in the soil that you're not trying to control everything. Instead, you're trying to optimize plant growth so that the plants can then do what they need to do to grow and they'll adapt. They'll take in microbes that, for example, if it's a bad environment, if it's a bad soil, if it's got too much salt in it or something, they'll take in microbes that help them to grow there. So if you grow the same crop there, especially if you could take your seed and grow them year after year, season after season, then they'll adapt to that soil using the endophytes that are naturally resistant in those environments. So it's exactly that self-optimizing system that involves plants, microbes, and soil, the soil community. And yeah, just following up on Harriet's concept there with something else.
Sure. And I want to play devil's advocate for a minute here, because I can also hear some people thinking, well, I've got crops failing out in the field right now. I don't have time for them to make these connections with endophytes and overcome whatever it is that's causing them, well, to fail, whatever that might be. And what do you say to that? I mean, how do you start to investigate if you don't have this level of understanding of crop health and soil biology to troubleshoot the system to make sure that you can still get a yield at the end of the season?
Well, now you're talking about something truly practical. I can't tell you what to do. I mean, there are situations where the environment is so bad that you're not going to be able to do anything, especially in terms of endophytes, to grow plants in that environment. So if it's a shocking drought, the drought is so deep that plants go, beyond the wilt capacity. No endophyte will help it, right? So you could, in those cases, I don't know what you do. Hopefully crop insurance will save the day, or, not, I couldn't give you advice on that if it doesn't work.
Okay.
Should it be the same question that you asked me, Oliver? The one about something's not working,
How nice is this?
crop failing, what do you do?
Yeah, Harriet, we were basically just exploring the practical side of whether the plants and the biology that supports them are able to respond quickly enough to the challenges of, well, the practicalities of working out in the field. If you're at risk of losing a crop, or if you're starting to see animal health decline, do you wait around for the system to self-regulate, to, I guess, bounce back from these missed connections or systems? Or are there ways to intervene, or at least start to diagnose or troubleshoot the system that could give you some insights on how to make corrections?
Look, that depends how cool you are. I recall I talked to Mark Shepard, and I was talking to, and I mean, he's a person that's really cool. He says, if I have a disease, what a bummer.
Yeah, we got you. You were talking about your
This is
conversation
really
with Mark.
silly.
We'll get her back. Until she comes back, I think I could probably fill in what the conversation with Mark was about, because he has the very well-known stun method, where if there is a disease or if there is a challenge in one of his crops, Mark often just lets them die off.
He just
His
lets
stun
it run,
method will
and
allow them to to bow out, and he just favors the ones that continue to do well over time and that showcase the characteristics of the vigor or the production or the other qualities that he's selecting for, and breed based on the reproduction of those crops or animals as well. And I know that's been a guide for my own farm, and that's kind of how we're approaching this topic as well.
This is kind of create land races, and that's the kind of adaptedness. But it requires really that a grower is able to grow their own seed season after season to do that. And if you're dependent on companies for seed, say it's hybrid seed, you can't do that. You're not supposed to grow those. So there's a certain amount of freedom that you have to have with your own seed, growing your seed, in order to actually build up the microbiomes in plants.
Well, this is kind of what we were talking about the last time we had this three-way conversation with you and Harriet, is that there is a huge amount of untapped potential in going back to community level seed saving, and eventually just promoting the genetic material that does well under the conditions, and maybe even the deficiencies or the pest pressure that you have in your particular area. Have you played around with that in any of your research?
We're not trying to create land races, but what we do is one of the things that we tend to do is to try to move endophytic microbes from one plant to another. So we'll do things like, for example, take wild weed, a weedy plant that we know is filled with lots of endophytes, based on microscopy examination and other kind of analysis, isolations and so forth. And then we take the microbes out of it, not isolating, but grinding up the plant so that the bacteria come out. And then we put those into, for example, grass seedlings, so that we can evaluate what their defects. We're actually trying to figure out how to move microbes from wild plants to cultivated plants, so we can essentially restore microbiomes in some crops. So that's the idea. But we do those kind of experiments, moving microbes around between plants to observe how it works. It's very, very similar to the soil thing, where others, for example, there was some work done by Rusty Rodriguez, and I think it was the early 2000s, where they actually looked at some plants, some grass plants growing, panicum growing on a cline from high or low salt to high salt. And they evaluated what endophytes were being taken in as it went through this cline, getting higher and higher salt concentration. They identified a fusarium, I think it was a fusarium species, that they could then take that and put it onto different plants and acquire the same kind of drought tolerance or salt tolerance. And they actually call that phenomenon habitat adaptive symbiosis. It's the idea that plants are getting endophytes that help them to survive in an environment, in specific environments.
Fascinating. I see many parallels to that concept, even transfers of the human microbiome to people who are suffering from gastrointestinal diseases, or, I mean, let's face it, making compost or bringing in healthy soil from a forest nearby is a similar concept of kind of inoculating something that has these present cycles and systems intact in them, and implanting them in an area where they're deficient to see if you can kickstart that cycle and maybe increase the speed at which that other damaged system can recover. Harriet,
Yeah.
do we have you back already?
I
She's-
hope so.
Okay, fantastic. All right, well, let's give this another shot. Go for it.
Okay, now I'm going to speak at double speed, to make up all this fuss. Basically, what we need to understand is that we have several components that need to meet. So there is a lot of talk about endophytes and microbes. There's a lot of talk about mineral nutrition. And what we always forget is that this has to grow into structure. So what we really need to watch and promote is soil structure. All these things can't work properly if we do not have gradients in the soil, crumbs, burrows, and all these things. So the thing is, and for me, it's better to be very imperfect in all of these areas than bring one of these areas into super perfection. So if we have an understanding where we say, for example, I'm this super plant nutritionalist and I know exactly at what phase of the plant I have to apply which nutrient as foliar and how to brew all these, yes, one box. Reduce that to half of the effort, take care of endophytes in the soil, and it's much easier. And the thing is that the more you can delegate to your system and to the system's robustness, the more relaxed you are as a farmer. And this is so hard because you have to be so brave to really slowly wean your system of your intervention. As I said, I'm not cool as Mark Shepard is. In some cases I am. I said, either you grow or you die, I don't care. And then if they did, nice. And if they didn't, okay. But if you really depend on your crop to make an income, it's hard to be so cool. And this is why relevant for farmers, small pilots. And also understand that seasons are not equal, and what works in season X is not going to repeat in season Y. I cannot tell you in these years when I've been gardening how often I thought, now I understood it. This is the way I'm going to do it, always. I've learned to put my eggs in several baskets, because especially in the chaotic climate we had in the last year, it's always good to have several, like seeding directly, preparing some transplants, different dates. And then one of all these things is always going to work. And if 3 fail, who cares? If you have the seeds and they don't cost you much, who cares? And this is back to the seed saving. Knowledge about seed saving and seed sharing rings and how the genetic side of it, it's really important, and to do good quality, because what's very often misunderstood is just you throw a bunch of rascals out there and they're going to do the trick for no, they aren't. You really need to be constantly selecting for what you need, leaving diversity in it, confronting them with new situations, bringing microbiome in, but really you need to look that what they do is what you want or what you need. So you're a factor in this system. And then the microbiome is, there are so many different classes. And for me, it was appalling to see how much fraud, actually, and I really want to call it like this, is already in bought mycorrhizal inoculants. if 80%, there is meta studies that have looked at that and say 80% of the mycorrhizal inoculants, they simply don't live up to what they claim. There is no association formed. Some of them have dead spores, some of them have pathogens, whatever. And I mean, then you're as a farmer, you think you're buying the bang in a bottle. It doesn't work. You think mycorrhizae don't work, but in fact, they were just dead or defect, or the system that you have doesn't support them because it doesn't breathe enough. So, or the plant is a dwarf plant and doesn't accept them. And now we become fashionable. We say, okay, we have the mycorrhizae, we have the microalgae, we have this, we have that. And you know, the spectrum is so large. And then farmers very often, they get confused because you think you can't do all of this. So my personal inclination is to do less from this perfection control pathway and go more to black box pathways where you say, I work with soils that I take from one part to the other. And I know and I trust that when a weed has grown in a place and it leaves its microbiome in this place, we know from experiments that always the next plant growing in this place will pick up some endophytes from this place and have them. So this is this succession. And that means, because I have just seen a bit of this question in the chat with the corner of my eye, of course, if we have wild plants, that is weeds, in this system, they bring stuff into it with them. And the question is, and this is the sore spot of all these regenerative ideas, is how do we deal with this? And of course, redefining them from weeds to caretakers of the soil doesn't solve your problem. That if they do their job too well, they just smother your crop. So these management things, how do I deal with weeds? And how do I deal with things that I regard as pests? And how much poison is per default in my system and how much compaction is per default in my system, that's decisive about your success on the microbiome side. And this is something that I find interesting because we talk so long about these things, about weed control and aphids and all these things. And compaction, and it's so boring. I mean, I cannot tell you how long Elaine Ingham talks about aerobic soils and decompaction. And you go out there and you name the field in my vicinity, they're all compacted. No change. So understanding this, and that we need 3 years to establish fully shared metabolic pathways in soils because the division rate is so low. 3 years. You do something today and you will see the full outcome in 3 years. 1,000 generations of microbes. So one way of speeding this up is to say, I incubate them a while together so they have a head start and a bit of a more favorable environment, like soft composting, not hot, colder composting methods. So they can habituate a bit with each other. Yes, it's a selection, we narrow diversity, all of this, but we already have a consortium that knows each other a bit. One possibility. Or else, of course, the foliar feeding hack is always nice. If you see that your system is going down the drain, don't wait too long. The thing is, what I know from experience, when you have set up diversity well and soil is kind of aggregated and you know you have done something for microbiology, in spring, in framing stage, one really loses faith. The plants start to grow and they just don't look good. You know, they grow and they're kind of a bit too yellow for your taste. And you're like, oh my God, and you get really nervous. And they don't grow as fast as when you really give them nitrogen. And you get really nervous. And if you know the system and you see that they are still doing their thing, still catching sunlight, and they're not limp and they're, you know, not really signs of unwell-being, but just of nutrient shortage a bit, that's okay. And that's a moment where you can say, I keep cool and I have the trust that this system is going to do it. And usually what you see after a couple of weeks is that microbes kick in and that they start to really green and go ahead. And when reproduction comes, these plants, they're just really pulled through, while these that you pampered fall off. But this is experience with the system, and I know how nervous you get when you see that. And I know that you really need experience with crop scouting to discern, do I have an illness? Is something going wrong? Do I have adverse microbiome here? Do I need to do something? Should I give a micronutrient foliar? Whatever. Or can I just entrust myself and say, this plant just really is eager? You know, it's like these cow's ears. If they're droopy, you know this animal has something. And if they lift their leaves and if they attract the sun, you already know this plant is feeding. It's on a good way. And then you can just kind of pimp the system, but don't interfere with it. So this is practical side of it. You need to train your observation. And this is a development, by the way, crop scouting. I just found accidentally crop scouting apps for greenhouses, for peppers and these things. And you have then your camera that runs through there and it just brings back the information, all you need to know. And it gives you better advice and better prognosis of your crop and everything than you could possibly do it. This is how it's marketed. And I'm sure that in many cases this is true because our crop scouting capabilities are really not trained. If you train your observation and you know where to look, you really can extract so much information from being with these plants, the way they smell, the way they move, the way the architecture is, because architecture has a high degree of plasticity. Back to James, epigenetic regulation of the genes through changes in amino acids in the plant, through changes in the mineral nutrition. It's just 100% immediately changes the whole metabolism of the plant and its architecture around and the behavior. And if you can read these signs, you know exactly where your crop stands and you know very well what you can expect from this season. So, of course, you can have the fancy gadget in this controlled environment that tells you the right thing, or else you learn yourself. And sometimes the hard way. I don't want to conceal that because sometimes you really make a mistake. Yes. So learn small.
Man, there's so many amazing nuggets in there that we could spend the rest of the time unpacking. But I do want to bring us back to this last question that I wanted to explore between the two of you, which is, and I guess I'll have to expand it now too. What sort of research or field trials would you like to see happen in the upcoming year in order to advance some of the concepts that you're interested in? But since you had also mentioned about crop scouting and the importance of observation, maybe I would add to that also what sort of educational programs or trainings would also be valuable to see expanded in this upcoming season. Maybe James, you want to take that first?
Yeah, I'll say what the research area I'd like to see. I mean, this whole, from my earlier discussion, this interaction, the genetic interaction, that to me is the new that we really have to understand. Understand first of all the mechanism. How do these microbes truly have this effect? That's the first question. And it looks like it now based on some circumstantial evidence, but do they really have that effect? And then the question becomes, how do they have that effect? And what is the manifestation of that? Are there more genes? Is methionine, there's some evidence that that these high microbial corn plants, for example, are loaded with methionine. And so their whole internal genetic architecture changes and these high microbial corn plants start throwing off mutations. So the other thing is that these bacteria actually colonize the nuclei of plants. So the possibility is that there's some direct interaction between the bacteria and the plant that alters gene expression in the plant. To me, that's the biggest question that I really am most excited about. I don't know if we're going to be able to answer it, but we're trying, and there's some other projects out there that have actually pointed this direction, that there's increased genetic expression in plants with high numbers of endophytic microbes and soil microbes. So to me, that's the biggest area. It's very basic, but I think it's important because it affects, crop adaptation to the environment and a changing climate. You want to be able to enhance evolutionary change in a plant in order to get variations that are going to adapt to the environment. It's like this self-optimizing system, right? I mean, that's part of the changes that plants need to make variation in order to readapt to an environment. Stress, for example, stress requires a lot of changes, and these endophytes, we know that they increase stress tolerance in plants, and it may be part of that may be a direct genetic effect in the plant. So those are areas that I'm interested in, brand new areas that might be important.
Yeah, that's super interesting, especially as we're all battling with a very fast changing climate. The idea that the crops that we plant could make changes or adaptations much more quickly through these relationships is really cool. And like you said, you may not be able to prove something, but that is what makes it exciting, right?
It is.
Harriet, same question to you. What would you like to see more investigation or field trials done on, or perhaps educational trainings?
Learning how to read your plants is certainly one that I think is must happen. Otherwise you do not know if you're on the right track or not, especially if you embark on this seed saving avenue. This is one thing. The other thing is, even faster than genetic change of plants, microbe-mediated or not, is okay. If we go back to the very root of these problems that we have, we need aerated soils and a soil sponge. And so the question is, what input channels do we have into these soils? And in general, what you read is photosynthesis, and between the lines is like plants are the only input channel in these soils. And I've spent a while just collecting alternative input channels into soils. And one reason was because in the old biodynamic literature, they have really conducted research where they've shown that bare fallow in winter, so this is the scary ghost of regenerative agriculture, bare fallow in winter was really giving good yields while building carbon pools in the soil. So this is something where we say, how can that be? And thinking of it, we have, for example, groups like microalgae in the soil, and cyanobacteria that even can photosynthesize with far-red infrared light. We have melanin in the soil that can absorb any kind of radiation and kick off electron transfer to microbes. And then we have synthesis facilitated from that input channel. And I'm sure that there's 50 others that I have not thought about, but nature has. So to allow our thinking to expand and go away from this single route to say it must be our crop plants that have to deliver some functions, and going and broaden our vision, there is so much metabolic crazy pathways that microbes can do. And the question is rather, how do we guide these things so we have construction instead of destruction in the soil? So we have building of biomass, building of structure, building of consortia. This is something that I would love to see investigated. For example, melanin has this fantastic side effect of being paramagnetic. This is every adept of Callahan is like, oh, we want to have this. The only way that we can get it is putting basalt in our soils. Nope. Annual lignin apparently can confer this thing also to soils. So all these pathways to get physical properties into soils are totally neglected. And I think if we would look after this a bit better, and this is really also immediate improvement of the system and this capacity of the soils to deliver growth, I really would enjoy that.
Can I add something?
Yes,
Harriet,
please.
that relates to what the microbes are producing, right? What the microbes are doing and delivering to the system and to the plant. But there is another thing that I think needs to be understood, and that is that plants actually can't grow properly without those microbes in their cells, right? So we don't, like, for example, we get no root hairs if you don't have bacteria going in. So this whole question, I mean, it relates to what Harriet's talking about, because some of those nutrients that are some of those things produced by microbes are like hormones, ethylene and auxin and cytokinins. And, you know, it is not fully appreciated that plants actually have to have bacteria inside their cells to grow properly. I mean, I'm not saying they can't divide and produce some cells, but they don't, those cells, those like root hairs don't elongate. You oftentimes lose behavior patterns like gravitropic response of roots. You get bizarre development if you don't have bacteria there. And then when the bacteria put back on the system, back on the seedlings, they grow quite normally. You know, so we know that there is this, if you look at leaves of plants, you look in the meristems, you see the meristematic cells, for example, tomato, they're full of bacteria in those meristem zones. They're the full extent to which plants actually are a whole community of microbes and the plant is not fully becoming more fully appreciated, but the details on how that works really are not understood. And that's an area that would be really important for people to work on. And it dovetails with Harriet's suggestion.
Yeah, it does indeed. And these are all fascinating things that I would also love to see explored and understood better. And I think that's a good place for us to open it up to questions from the audience. So go ahead and raise your hand digitally there, and I'll call on you in order. And let's make the most of the fact that we've got two fascinating researchers here to answer your questions. Don't be shy, whoever wants to go first. Jose.
Hey, thank you so much. So I have two questions. Hey, how are you? The first one would be for James. So I heard you speaking about seed saving in a podcast, I think. If I'm not wrong, you had some suggestions on ways to save the seed that would help the seed to keep the endophytes. And so what would those be? Do you have any suggestions for people that are saving seed? And actually, I just want to say something that I'm fairly new to this, but that a lot of people that I've met in the seed saving circles, especially in cereal, what they do is to prevent passing diseases from one generation to the next is to treat them with copper. So this is a preventive mechanism, but that in my view, it will kill a lot of beneficial things. So I wonder if we could, instead of this, to add more beneficial microorganisms, if as a seed treatment and a way to get rid of diseases that the plants might have. So yeah, the first part is my question. I have another for Harriet, but maybe I can say it after. I don't know.
Hey, let me answer this because you'll confuse me if you ask the next one. So let me go ahead. We think that actually the reason we have so many problems with seedling diseases is because we don't have a a complete microbiome on the seeds. And if you consider the way that we typically gather seeds, we produce them, we collect them at the earliest point, we take them in a dry place, a dry and cool place, which restricts microbiome development, right? Normally what would happen oftentimes is seeds would stay outside and there would be rain splashing, there'd be changes in temperature and moisture, and so the microbiome essentially matures on the seeds and in the seeds. Okay, so that leads to a loss. It may not be how critical it is, I can't say, but a loss of components of that microbiome, and leading and typically when that happens, then you see more seedling diseases, things like damping off, fusarium, rhizoctonia, like that. And so you have more diseases, and the solution, we think, and from our experiments, has been you add microbes, not try to sterilize the seeds more. Actually, what we see when we try to sterilize the seeds is almost invariably seeds become weakened, and fungi that may be there as an endophyte and having no negative effect on the plant then begins to have a negative effect. So yeah, this ultimate solution, we think, is adding microbes to the system. And that's where we go back to our experiments trying to move microbes around. You can't isolate most of those microbes out there. I just heard of a recent study where 9 out of 10 cannot be cultivated and are largely unknown. There's only a few, that are, and so we're trying to find some way to move microbes without actually isolating them between plants so that we can conduct our experiments.
I would like to add just one sentence, and this is from my experience: when you inoculate seeds with microbes, it becomes digital. Either they grow better or they just degrade. So especially when you have very old seeds that are of bad quality and nutrient deficient parents, and then you put a lot of microbes on there, they just digest it and they don't even attempt to germinate. So just buy good seeds, work with high quality seeds, and inoculate them. And what I just learned this week is that there's now a product that is not mycorrhizal fungi, but an endophyte in Europe, seed forward, and it's a Sebatzina, it's an endophyte, as a seed coating. And I was really thrilled by that, by cutting edge science, because this is not so tricky to handle as mycorrhizae are later on, and you can really just as well add it as a seed coating.
Just to, can I just add up and ask the question to Harriet quickly? And so would you have any, James, by the way, any suggestion, or Harriet, for on the physical way of storing, like with more moisture, say? Would you recommend that? Well, not too much, or not to germinate?
Could I say that, Harriet?
Of course.
My feeling is, yes, you want to mimic nature as much as possible. That's the best way. But the issues are, you have to be careful with I'd say if you have some substrate that has microbes in it, for example, some animal dung or some soil that you can mix into the healthy soil that you could mix in that's not too moist, right? You can mix in there. You may actually enable the seeds to acquire more microbes from the environment. But that's an experiment. I'd say you could try something like that. It's a suggestion. I haven't done it. Harriet, you want to go ahead?
I think it depends very much of what we're looking at. For example, what I used to do is in these fermenting fruits, when you collect microbes from there, it's really interesting. If you have plants in bad health, these jars where you ferment it, they stink as hell. And when the fruit quality becomes better, the plant is of better health and the microbiome is better, usually what happens is lactic fermentation and they don't stink anymore. So what I used to do is when I started off with the system, I used to add yeast, baker's yeast. So you have just this ethanol in this and it kills off the microbes that the plant brings. I stopped that just so that the plant just really passes on from generation to generation what's on it. And the other thing is when you ferment, that's a chance to bring in other microbes. For example, I very much work with these 6 biodynamic herbs because I know from experience that they're super plant helpful and beneficial. So you can add them when you're fermenting or when you're seed bathing, or seed bathing with milk, for example, in cucurbits. Very good. And these are some tricks to trigger off. I found that cucurbits really fancy this initial pulse of lactic fermentation around them. Very healthy plants, very sturdy plants. But storage, this is a tricky thing. What we in general do is overstore seeds, store them far too long. So in general, what I see is in many crops, it's not all, some get better in the 2nd or 3rd year, but it would be really good to really save and use. And you see that when you overstore plants, what they usually do is they start to, the first foliage is very narrow. And that's a signal that the hormonal balance is already off when you start. As James says, you can still in this stage, when you inoculate that plant, even after germination, when you fertilize or when you water it and you have just a dash of a bacterial solution in that, will still help the plant to settle in, and you see that it starts to make normal shape foliage and everything. So it's never too late. But in general, to have an easy life, good parents with good mineral nutrition, I'm not talking of macronutrition in excess, is enough to bolster them out so they can make biomass if it has been short, but tend to back off this, and fresh seeds. And then it's a question what you can do. For example, I know the first person that I've talked to that has no blight in tomatoes, he said, well, I have no blight in tomatoes. I said, what do you do? So what he did was he left the tomato outside, whole overwinter, the whole fruit covered under just leaf duff. And then when in spring, when they started to germinate outside on the spot, then he would take the plantlets inside and expand them so that they have this head start that they need. And I found that really, really interesting because this way you do not break this chain. You do not have this drying moment in these plants. But it's very hard if you're operating large scale to do these things. But think of the logic behind it. Just really fresh and strong seeds and a good inoculum.
That's super good. Thank you so much, both of you.
Thank you,
Well-
Jose. All right, let's move to Jess. Go ahead.
Hi, thank you so much. This is kind of blowing my mind. I have a few questions. One was to James. You were talking about the bacteria in the plants being in the DNA, if I remember correctly. I mean, this is very advanced stuff you're talking about. So my question was, can the bacteria from one, if it's growing alongside another plant, can it jump to the other plant and be absorbed and become part of the other plant?
The answer is yes. The simple answer is yes. That's what happens. In fact, that's what we think. That's how we think it works. You know, with cover crops, right? Cover crops have different endophytes in them, and there's some of those endophytes are going from the plant into the soil, and so you have these plants essentially sharing endophytes and other beneficial microbes, right? So that's what happens when the more cover crops, the more beneficial endophytes you have going into the system, sharing. And the other question was, now you said, into the DNA. Just to clarify, the bacteria go around the nucleus, and
Right.
they actually get trapped in the nuclear envelope, but we think they're also going in the nucleus around the DNA. And we think that's happening because the rest of, once they're in the cytoplasm, the plant cells degrade them, but in the DNA, they don't degrade them because it would destroy their DNA. So we can see these bacteria accumulating around those. And it's very common. We think it's how the plant moves their endophytes, certain bacterial endophytes from cell to all their cells when they produce it, because it's growing in the meristem and those are dividing. And when they divide the bacteria, it divide too. And they end up in that nucleus when it reforms, and that's not so critical for the leaf cells and cells like that. But in the pollen mother cells, we have the same thing happening. And so we think that the actual DNA changes could be happening in there. And it's interesting to explore that idea because it offers whole new explanation for stress tolerance in plants and some of the other phenomena that we're seeing in endophyte infected plants. And it's so important for, breeding new plants for people, right?
That's super exciting. Thank you. I asked that question because I've got one field of wheat at the moment with Oxalis pescapre, which is invasive plant. And I was kind of looking, okay, what are the benefits of this, right? Because I'm always looking at like what these wild plants are giving. And one of the things I read about Oxalis pescapre is they've discovered that it has a bacteria that stimulates plant growth. And so that's really exciting to think about that. It's like, okay, so maybe that's jumping to the wheat. And also that they have this, well, anyway, I tend to look at the like not to interfere. If there's weeds in the field, then they've got a function. So I have another field with calendula. It's making sulfur accessible. It's creating a beautiful understory that's humid and covering the soil. I'm in the Balearics, so we have a lot of sun that dries the soil out. So yeah, I think like I'm with Harriet on that one, and like let's be messy farmers.
You're thinking like an ecologist too. What are the benefits? I mean, a weed is a cover crop, right? It's another natural cover crop that
I
is
didn't pay for it.
donating microbes. And you said already, people have isolated microbes and they found that those bacteria or that bacterium, whichever one they focused on, was also growth promotional in other plants, which suggests that they're sharing those microbes, in the soil too.
Yeah, absolutely. And when the field gives you thistles, well, it's a free cover crop. Great.
Yeah, you have to be careful. Some of them might inhibit. Got to make sure you don't have an inhibitory microbe coming off of that weed too.
Right. Okay. Thank you very much.
You're welcome, Jess. Thank you.
I'm just using my pole position today. James, I just had an idea.
Okay.
Do you know much about transfer of beneficial microbes through leaf-sucking insects? Because we focus always on these insects as vectors for the nasty ones, but there is no reason why they should not be able to transport the beneficial ones as well. I cannot recall that I've ever seen anything discussed in literature about that, like aphids transporting other beneficial bacteria.
We do know that some aphids will transport microbes in their bodies for one thing, but also if you ever look in an aphid in these galls. We were studying aphid galls several months ago, and in oak and sumac, and these galls actually, if you look at the tissues inside, of course they have the young aphid in there, the larval forms of the aphid, but if you look at the tissue bordering the aphid that they feed on, it's actually full of bacteria. More bacteria than you see in those other cells. So quite possibly those bacteria are there to provide some nitrogen, likely for those developing larvae. And if you look at the larvae, when we look at the larvae, we could see that many of those bacteria were wedged on the scales or on the body and in the body because they were feeding on it, going into the bodies of the larvae. So they're carrying those bacteria around. Now that's a symbiotic association, right? So, but it's a symbiotic insect bacterium and bacterium plant. Of course, the tissue that the plant tissue that grew is that gall tissue,
Deformed, yeah.
deformed gall
Yeah.
tissue, but it's kind of a growth promotional, likely a bacterial mediated growth promotional effect result. Gall.
Next year, I'm going to pick a gall, blend it up, and see if it promotes growth, huh?
Okay, try that. Try that.
I will.
It's a good idea. Yeah,
Nice. Let's go to the next question. Siger.
Yes, thank you. I'm Siger. I'm from the Netherlands. And more question to Harriet, I think, regarding seeds and seed saving. I mean, the more I read what you are saying and the more I read about it, it's like a total no-brainer that seed saving is crucial and necessary. However, if you want to do it as a farmer and you do it in the Netherlands, you're illegal because you're not allowed even to do your own seed saving. And I think it's in a lot of the EU law at the moment, I think, is not allowing any seed saving. So
No.
what is the right way to still be able to do this crucial thing and not, as a farmer, get into huge problems?
No. The correct status, as far as I'm informed, and there may be another twist currently, but the correct status is that you're allowed to save your own seed and also use it, but you're not allowed to trade it
As far as I
as
know,
a farmer.
as far as I know, I just share the database in the comments. In the Netherlands, you're not even allowed to save your seeds.
Wow.
So, and that's in, I think, if I look quickly in the database, like 30 or 40 countries of that database of 120, 130 countries, you're not allowed even to save your seeds. I mean,
Wow.
you're in deep trouble if you can't do that. What do you advise farmers to do then? Because otherwise you're in the hands of the big five and they only provide you the seeds that are produced without the right microbiome.
That's a hard one. That's really a hard one. I'm not sure if you could ask for experimental status for research, maybe. Maybe there is a loophole there. But then I doubt that you can sell the product.
Yeah, exactly.
Yeah.
I mean,
that's really bad because I know in Austria it was even you can get the permission to have it on your farm as a locally registered variety. But that's really bad position. And I know what shocked me was when I heard from that last round of legislation in Brussels that they said they would never be able to get a law through against the pressure of big ones.
Yeah.
And I was like, 10 years ago, nobody would have dared to mouth this even, to say that we cannot make a proposal for a law because it would be thwarted by this party.
Yeah.
So this is really bad news for me. I didn't know that.
Yeah, I mean, look at the database that's in the comments. Yeah. I was shocked when I read it.
For me, ask Vananda Shiva. Really, I mean, she's a lady that can make Vananda Shiva.
Oh yeah.
She has made a record in India, and I think we need this record here because this is, for me, this is a human basic right to be in charge of your food and of your seeds because this is, the source of all.
Yeah, exactly. Yeah. No, so yeah. Okay. All right. Clear.
Yeah.
Thank you, Siger. Lorenz, go ahead, bud.
Yeah, maybe a follow-up comment and question. I know that in Germany it's allowed to save your seed on the farm, but you have to pay a fee to the governmental authorities. I don't know exactly which one, but this is definitely allowed in Germany so far. But I'm also wondering in that regard, because I learned in farming school that basically if you continue breeding your own variety on your same soil, and I also seen it on our farm, the vitality of the crop of this variety slowly decreases. And I think it's due to degraded soils and not the perfect microbiome. Do you think that if we manage our soils properly, actually the opposite will occur and we get better vitality and better yield by breeding in regenerative soils? And maybe this might be a solution for big scale farming to have regenerative feed companies where they breed and with an intact microbiome and sell those seeds in a bigger quantities.
So for me, this is something I just answer quickly so we can head back and have the best value of James. Basically, the most important is start with a large, either very well adapted variety and really maintain it well, one plot. The other one is make a wild mixture so you have a lot of diversity and keep diversity in it. And then it's really, it's a tango, because what you do to it is what it responds to. So if you constantly jump around it, it will adapt to this pampering. And I know what I'm talking about. And the main mistake that one usually makes is that either you win it too quickly, you start with a variety or a genetic germplasm that you demand too much, that it cannot fulfill with the mineral nutrition and the microbes that it has. And then it just really goes down. And the other thing, never forget, if your soil is crap and you try to breed a variety to this crap soil, really compaction is the killer. You demand so much adaptation. And then you have the backpack that this crop had in terms of micronutrients and everything is just lost in some generations. Usually it takes 3, 4, 5 generations. And then you see what you really have from your soil, from your nutrition, then your eyes are opened to what you're really doing. Because then either it goes up or you lose it. So genetic diversity, large enough populations, a good selection, strong parents, and in the beginning, really keep levels up with micronutrition sometimes. And always have your backstore, from some years ago where you can fall back and say, okay, now this development has totally gone in the wrong direction. I start. And as I say, these systems, they need exchange. So back to James.
Okay. So I saw a note popping up only for a brief instant, and I agree with the statement, and that is, and I think Harriet she and I would have, I think we talked about something similar a couple of years ago, but the idea that you can't just take any variety and grow it regeneratively. You can, but they're not really adapted. It's not really adapted to it. It may have, for example, some of our corn lines, for example, may have lost the nod genes that enable to connect to mycorrhizae, and there may be other losses, that these corn plants have had. And then you have the fact that when you get the corn you buy, they're hybrids, and you have a certain hybrid vigor, and we don't really know what that is, but after if you grow that plant for the next cycle or you keep breeding it, you're going to get outcrossing. And so it's going to genetically change. And so that may be non-optimized too. In other words, really the variety, you need to get a variety that's good to be grown regeneratively. And that's my suggestion. Harriet, did that sound like something that's worthwhile?
Yes, absolutely good. This is what we've been talking about. There is some, even hybrids, there is some hybrids that pick up microbiome immediately.
Yeah.
You just confront them and they make wonderful root hair and they grow perfect as if they had done it all the time. And then you have some, they're just, dumb. You just confront them and there is no interaction, no root hair, no nothing. And they just sit there and wait for their food. And you're like, you're not going to get it from me. And they can't acquire it. They don't work. And this is an old wisdom from breeders. They say, breed in the environment if you want to see the result. And this is where these varieties show up and produce, while the other ones fall off against them.
All right, I think we probably have time for one more question. Frederick, come on in, Frey.
Hello. James, I have a question. You mentioned putting some soil with the seeds for getting more microbes in. Are you thinking about using the soil from the rhizosphere of the plant where the seeds come from, or are you thinking about the soil where we would plant them? And there's a second question. Should we let the seeds go through the climatic changes of fall and winter? It's a question for both, by the way, because Harriet knows a lot about rhythms and microbes.
I would say yes. Go through the cycles. You need to do that. It's natural for the plant some plants require that. For example, stratification, right? Some cold cycle, period of cold. Okay, so yeah, you have to do that. In terms of exactly where what kind of soil, could be rhizosphere. I mean, I'm thinking just some microbially rich soil. It doesn't have to be from the place it's going to be planted from, but it could be. I mean, that might be the smartest thing to do, to begin adapting the plant to the environment. So yeah, I mean, I would say do an experiment and try to do it multiple ways. Did that help?
Yeah, that helps. Or maybe mix them both.
Yeah, mix them both. Exactly. Try different things.
Mm.
Yeah.
There would be no germination issue with the rhizosphere because there you have the active microbes still working with the roots.
You could have germination issues anyway. Either way, I mean, there's going to be fungi in there, so the fungi could, overwhelm your seeds. So you have to kind of lower the amount of moisture to a point where you're not going to have overgrowth of other fungi. So you'll have to play with it probably, Frederick.
All right, thanks.
You're welcome.
Harriet, did you want to add to that as well?
Oh, but I'm curious for Lauren's question. If
All
we can
right.
squeeze that in, I'm saving my answer.
Yeah, you're a favorite, Lorenz. Go for it, bud.
Thank you. James, one question to you. I'm wondering about the anaphytic transfer. Is it possible, in your opinion, to also achieve this in a later stage of plant development? Because, for example, now in the coming season, we will plant in springtime our grains, but there will be not a lot of material to harvest anaphytic microbes from eventually. So I was thinking if it's possible to apply them maybe with the first foliar spray, and if it's actually possible that the plant can take up the endophytes also through the leaves, if there's a lot of other goodies in it that the plants really like to take up through the leaves.
Yeah, the foliar works. Grasses might be better than some other crops for foliar, but all the entry point is in the meristems. So, wherever the flowers are developing, wherever the inflorescence is developing, where the growing leaves are coming out. So when the microbes get on there and those developing tissues around the meristematic tissues, some of them will go in, some of them will colonize on the surface. So absolutely, you can do foliar. The other thing you could do is, even if you're putting on the root, there's one study with grape in which a yeast was placed on the growing root tip of the grapevine, and it took a number of days, but eventually it was on the fruit. So it moved from roots through the plant. The same thing has been recorded for movement from the leaves down to the roots. It just tends to take longer when it's going from leaves, from shoots to roots. But yeah, you can put them on at different times.
And about the quantities, I mean, I'm thinking about like going out, gathering some grasses that are in the same family as my grains and making a smoothie out of it and applying it with a big spray broom. Do you have any suggestions about quantities?
Well, I would put plenty of the juice there because, in the ground liquid, the concentration is going to be lower. So you want it, are you going to take seeds or are you going to take leaves to do the grinding?
I think the leaves.
The leaves. Okay, if you take leaves, then just make sure that the liquid you have is greenish, right? So you can see the chlorophyll there. And then you germinate your seeds in that liquid, or germinate them even on a paper, right? Or just take that and pour it into a soil environment. It might be better to sterilize that soil so you remove the competition. But you could do that just or not. Don't sterilize them. Grow them on sand. Germinate them on sand or something with low nutrient environment, and then flood them with the juice containing the microbes.
Yeah, thank God our soils are mainly sand anyways.
Okay, yeah, that's good. That's good.
Mine as well. Harriet, you wanted to jump in there?
Yes, I would like to just add a remark to this. We have these mental boxes, and we have endophytes, and these are good and we want them and they resist to come into our system. And then we have these nasty pathogens. They're just sitting there and wanting to invade our precious crops. This is a mental box. Basically, we're looking at the same organisms, and this bacteria and fungi. So as soon as there is contact, the plant will pick up some. And if there is vectors like insects and stuff, they will move around in the system. And the more alive the system is, the more self-propagation and transfer will happen. And this idea that we have, I must put it there, is just true for a system that's at the beginning of this regenerative journey. So if pathogens can spread, endophytes can spread.
That's a
That's
good
right.
reflection for us to perhaps wrap up on today. I can see that this conversation could continue indefinitely, but we'll wrap it up for this session. I want to thank all of you who took the time to be here, especially Harriet and James, for all of your contributions. It's always fun to get you two together and dive into these topics. I really hope we can do this again soon. Shame we missed out on David this time, but I hope he gets better soon, and we'll be sure to include him in a discussion in the future. And before you guys go, keep in mind that we're going to be having these panels every month now on different topics that are relevant to our community discussions in the climate farmers community. And if you or anybody that you know would benefit from bouncing ideas off of other farmers in your peer group, or wanting to learn from people out in the field, you can go to climatefarmers.org and sign up to our community for free if you're an active farmer anywhere in Europe. Yeah, that's it for this session. I really look forward to seeing you guys on the next one. Thanks so much for being here. Have a great rest of your night.
Bye-bye.
Thanks so much,
Bye-bye.
guys.
Bye, James.
I heard it. Well.